question marks are, again, around number of samples that you will get out, as well as amplification efficiencies. But it's not, it's not a deal breaker. The system doesn't completely break just because you don't input 100,000 cells.
Yeah, okay, great. That's good to know. I don't have the numbers in my mind right now, but I think I was using the version 1, and I think the requirements were even higher there. And yeah, I had much lower, a bit lower input, and it did work fine,
Yeah.
I think it's the requirements are lower now in the V2, right? I don't have the thing with me right now. I'm not in the at work right now, so
nobody is.
I think you changed that already, I think, in this V2, right?
Yeah, V2 comes with slight improvements, as well as changes in the user guide. The sample requirements have changed as well. It's really the volume that has changed. The concentration itself of the sample of its cell suspension remains the same, which is 3,000 to 4,000 cells per microliter. But now, instead of requiring 100 microliter of input, we only require 35 microliter of input.
Yeah, okay.
And so that translates to 35 microliter times 3,000 cells as a minimum is roughly 100,000 cells. And that's mostly attributed to the new cartridge design that we have. If you have noticed, the reservoir that accepts your cell suspension is slightly slanted, so that if you pipette a small volume, that small volume is on top of the inlet where the instrument will draw the volume from. And so there's minimal risk that the small volume will not actually be on top of the inlet port that needs to be covered by the volume.
Okay, thank you.
I see another participant, Romain. If you want to unmute yourself, if you have any specific questions, or maybe you want to introduce yourself first. I can still see the crossed out microphone, so I think you're still on mute if you are talking. Let me see if I can unmute you. I cannot.
You can always send questions through the chat.
Yeah. So Romain, if you have difficulties connecting with the audio, please feel free to use the chat window. So I'm just going to share my screen here really quick. Is it? So if you go to the chat window, you should be able to ask any questions through text if necessary. Otherwise, while people are waiting and calling in, just as a reminder, I think that's known to most of you on the call. We do have a knowledge base that we are currently developing and building out that should be a very useful resource for customers using Mission Bio's products. You can access it through support.missionbio.com. And let me just minimize that window here. And it is right now categorized based on experimental or the experimental step along our product line, starting from sample prep all the way to analysis. And in each panel, you typically, when you click on it, you'll be able to find either user guides or FAQs or other useful resources, including videos, to help address any questions you might have. What's super useful, I believe, are the FAQs really, as they touch on all kinds of different questions that customers have had most frequently in the past year. So we collected all of these and provided answers. So feel free to, as you work with our products, to consider this platform as your first line support to to support you independently. And we are about to. This is version 1. If you will, version 2 will be launched in a couple of weeks. That will improve user experience, as well as come with additional useful content that relates to more of our newer products that we just launched over the past 2 quarters. Okay, let me just get rid of Slack here. Kirsten, do you know, I guess, just out of curiosity, how long or how Switzerland is going to deal with COVID-19? Is there any shelter in place that is dated?
Well, I don't think I'm the expert in this one, but I mean, we still haven't reached. I mean, it doesn't look like it's going to stop very soon. We've had like roughly 1,000 new infections every day. Well, that's the official numbers of the health ministry. So I have no idea. But yeah, probably will take.
So ETH, the genomics core in Basel is closed right now.
Oh, well, we keep functional, but we have priorities now too, and we have to see what we might get involved in COVID projects. I have no idea. This is not, you would have to talk to Christian Beisel, my boss, to get more information about that. So I don't know.
Yeah, no, I'm just curious because some institutions are closed without any exceptions, and some institutions handle it with a tier system with high priority versus low priority projects, and then some institutions are still open, business as usual. So
well, the rest of the building, people, they're basically, it's kind of closed, but we have to keep functional because we kind of do something. We have to be ready for some kind of social service, or whatever this is called. Yeah, we have to be ready to help out, basically.
Got it. Thank you.
As the tests are actually running out on the market. So, but I don't know the details, so I'm sorry.
No worries. I'll give it one more try. I see Romain is on again. So I don't know if you can hear us, Romain. I'm unmuting you, but it's not working. So feel free to use the chat window in order to ask a question or leave a comment if your audio does not work. Okay. And then have you had a chance, Kirsten, to work with, even though you haven't really been able to produce any data yet, have you been able to take a look at Tapestri Insights already, the secondary analysis tool that we have to work with demo data? Or is that something that you reserve until you get a crack at your own data?
Well, no, we haven't processed any data yet. So I mean, I had a rough look, at how the the pipeline after looks like and stuff. But yeah, we haven't done anything with. We first actually have to sequence. So there's it's all just theory. So at this point, and we don't have. We first really need to process more samples, which is right now just not going to happen, I think.
Yeah. And I guess maybe that's also in the balance, depending on the results of the first data points that you will generate. Because right now you work with a commercial panel, well, with one of the catalog panels, and I don't know what your specific project is going to be, or if you are mostly working with client samples that are going into the core. If you have any visibility already in terms of potential custom project or custom design projects.
Oh, well, again, this is a question for my boss, I think. As far as I know, there is a project in the pipeline, but I don't know if this would be more custom, or maybe it's changed again, or, you know.
Okay.
Yeah, I don't know if there would be a custom panel, or probably, I don't know. Yeah.
Let's see. Oh, Romain.
Hello?
Hey, hello.
Hey, okay. I'm sorry, I have trouble with my connection.
Oh, no worries. I'm glad we can hear you now.
Oh no, I can see you. Hello, so I'm working for Raphael Itzikson. I don't know if you...
Yeah.
I'm in a French team, Saint Louis team.
Yes, Saint Louis in Paris.
Saint Louis in Paris, yes. And I am, in French we say like Master 2 students.
Mm-hmm.
And so I will be working on a single cell project. So I thought I might attempt the video to try to learn some things, but I'm really not expert yet on the subject.
Okay, that's,
Okay, yeah.
that's perfectly fine. So are you? You'll be. So have you? I guess my first question is. Have you already processed any samples or reviewed any of the user guides or documentation? Or do you have any specific questions, or is this more of an overview that you would like to?
We haven't received all the furnitures yet.
Oh,
so we haven't processed any samples yet. With the genotype plus phenotype approach, but the team has already done some single cell experiments, but only with the genotype approach.
mm-hmm.
I don't know if the meeting today is only about the genotype plus phenotype, or it's about all the approaches of Mission Bio.
This is about everything. This is really support team.
Okay.
Ask us anything. You
Okay.
know. So yeah, there's if you have a specific questions about the protein, we can we can supply.
Actually, yeah, I have a question. I'm actually like selecting the patients.
Uh-huh.
And are all the FLT3 ITD duplication covered by the AML package? I don't know if you can answer, or if you can tell me who can answer this question.
So it's a good question. So so ITDs, as you probably know best, are extremely challenging to detect using sequencing-based technologies. So most of the time, they're being detected using qPCR assays. Now, knowing that difficulty, we at Mission Bio have incorporated a custom algorithm that runs alongside with our standard genotype caller, GATK, at the level to call specific ITDs, internal tandem duplications in the FLT3 locus. The catalog panel, AML, consists of 5 FLT3 amplicons, in which 2 are known to have ITDs inserted. And so the custom algorithm will specifically assay those 2 amplicons for any potential insertion in triplicates. That's typically how ITDs are being incorporated. So I don't want to say that we assay all of them but we assay the regions that are most prone for insertion sites. So I can, if you haven't already, you can take a look at, if you know the insertion site and if you know organally where to expect ITDs, it's worth for you to review the BET files of either the AML panel or you are working with the AML panel, or are you working with the custom panel?
AML panel.
AML panel, okay.
Yeah.
It's worth then reviewing those BET files and then cross-referencing them against your orthogonal data. And if they're inside that region, then you should be able to detect them. Another point I want to make in that regard is that we have a few customers that then also revise the sequencing chemistry. As you may know, we typically recommend sequencing with 2 times or paired end 150 base pair cycles.
You say 150, right?
Yeah, 150
Yeah.
2 times. So paired end.
Okay, yeah.
Paired end. However, if your ITD, because some of the amplicons, even though they are optimized, the length of the amplicons is optimized on average to be compatible with paired end 150 base pair sequencing, some of the amplicons will have a gap in between. So particularly in the context of ITDs, if your ITD is at the end of one of the reads, read 1 or read 2, it's worth sequencing with 2 by 250. So our platform, our bioinformatics pipeline is also compatible with this type of sequencing output. It's more cost for you, but it's worth if you expect an ITD to be within the end of the read or potentially even spanning a gap region of read 1 or read 2.
Okay. And so if I'm not sure about one patient, who can I ask for validation to tell me it's okay, it will be covered or not, if I have a specific question about a specific insertion?
Yeah, you can always reach out to support. Sorry.
Okay.
support@missionbio.com, and Hema Fuerte, he was on the call as well. She's our colleague in Europe, so she's your primary point of contact for this type of question.
Okay, great.
And I can, so one point or one sort of source that you can access is designer@missionbio.com, and that's where, or actually you should really get an account on portal.missionbio.com. That's sort of your go-to shop to have all the bases covered with respect to software. Once you have an account, you can then, through Designer, by clicking on Designer, you can access the catalog panels, AML in this case, that you will be working with, acute myeloid leukemia. And then just to give you an example, obviously the data sheet that you can download contains all the specs, but for this particular question you have in mind, you can click on the download button. And then download the BET files. So if you download that, click. I already downloaded it a million times probably. So it's a zip file that you unzip, and it'll contain